Well, Dr. Andrew, please tell us what are you presenting here at this course? Which tools are you giving to the students and the participants?
Okay, so in this course, it's very different from what I've just spoken about. With UniProt, I'm actually now concentrating on importing and better representing the genetic variants that alter proteins. Okay. So what I mean by that is that you have a genetic variant that changes an amino acid or some amino acids or truncates the protein quite substantially, and giving an overview of what we have in UniProt currently, and then how you can use that information to further understand the consequences of those types of variants. And in this second day, really, what I'm doing is I'm broadening out everyone's ideas and understanding of how a variant, simple nucleotide changes an amino acid, can then change the whole concept of how a cell works in terms of its cellular processes, disrupted pathways, etc., like that. And then also trying to then bring back into the therapeutic side of things. So where you have a disrupted protein that's in a pathway, is there another protein that already has a drug that could be used to influence or affect the disrupted pathway?
Well, so let's start talking about those pathways and about the protein, how a protein works. Let's say that is more famous, the genetics, but not the proteomics. So tell us about the importance of protein in human body and in all living organisms.
Okay, so proteins, we are made of proteins, really. We're not made of genetic material. We are proteins. Our cells are fully made of proteins. So, and proteins are essential for the communication between cells, the regulation of an individual cell, how a cell differentiates into the different cellular types. And so where you have these changes in pathways upregulated or downregulated because of variants, this can have quite drastic effects on that cell or that cluster of cells, which then manifests into physical phenotypes, which we see. So classics are cancer when things go wrong. Other classic examples are Alzheimer's disease, degenerative diseases such as those, or developmental diseases, some of the ones I've been talking about today, where, at the embryonic level, things are not developed correctly, so synapses are not connected correctly, and then, the patient will come along with a phenotype of mental retardation.
So all our diseases, or even the way that we look, it's the center of that discussion is protein. And how the genes express those proteins and how those proteins develop in the body.
Yes, that's a good way of describing it. I mean, it's a genetic change, but it's then how that is physically expressed in the protein, and then how that protein interacts in the broader context of the cellular processes, which then manifests into the real disease. Your gene is not the disease; it's just the memory that's gone wrong, the hard drive, the book.
The way that it's read
has gone wrong. It's actually in how that is transcribed and how that physically works within the cell that then you really have the issues that you see. And that's why understanding proteins is so much more is so important, because knowing the genetic change does not give you the story about exactly really what happens within the cell.
Dr. Andrew, what are you working currently in investigation fields, and what the world is currently focusing in this field to understand what are we trying to understand in this moment?
With UniProt, what we're now starting to look at is much more taking this fundamental data that we have on variants, information that we have on protein-protein interactions, and bringing in a lot more clinical information. So you'll have a greater clinical context, so that there is this proper bridge between the clinical view of a disease and the biochemical view of a disease.
Okay.
And, allowing clinicians and researchers to be able to communicate much more cohesively in the same language.
Is the environmental issue, or the places that we live, the places that we visit, have something to do with the change of our proteins?
It can do over a long term.
Okay.
I'm only here for 2 weeks. It's not going to make much difference to me. But yeah, long term, if you change your environment quite significantly, the way your body reacts to that environment certainly definitely changes. There is, out of the context of what I'm talking about today, your gut microflora changes over time.
Yeah.
So if you go to a brand new environment, you're going to introduce completely new gut microflora to your system, which changes how your entire body reacts to the environment, is within how it plays within the environment. So there, you do get changes like that in different environments.